Professor Gary. We're all ears
Okay,
and
so
ready for you.
thank you. So I'm very happy to be here and represent the immunology crowd. I am sorry that I wasn't able to participate earlier and hear all those wonderful talks on neurology. And I have to say, I feel something like an interloper because my background in neurology is very limited. For the last couple of years, I've worked at AC Immune and have been on a steep learning curve to understand the challenges facing the development of products to treat neurodegenerative diseases. So if you'll permit me, I will share my screen. There are a few slides that I can go through to set the scene a little bit and hopefully explain a little bit about what's happening in the field and what we are doing here. So my first job, I guess, if I have to put it that way, is to say that AC Immune is a public company, and in my role as investor relations head and corporate communications, I have to say that we make what we call forward-looking statements. That might be an unusual expression for most of you. That just means that if I say something that refers to a possible future event, you shouldn't rely on the accuracy of that comment, simply because none of us, as far as I'm aware so far, none of us are able to tell the future. So that's what a disclaimer on forward-looking statements looks like. But then to get down to the subject matter, neurodegenerative diseases represent an incredible challenge for medicine today. And you can see the first point on the left-hand side there, the cost of managing and treating people with dementia is probably over $1 trillion annually. And that includes direct therapies, but also then all the ancillary costs that come with managing patients and care costs and so on. The most common of all the dementias is obviously Alzheimer's disease, and there are probably actually over 100 million people worldwide who suffer from Alzheimer's disease and who've had some kind of confirmation of that diagnosis. And you can see on the right-hand side the pie chart. Let me activate my laser. So you can see there's roughly 100 million patients who've had some kind of diagnosis. And that could include clinical dementia, and it could also include prodromal Alzheimer's disease. And by that, we mean someone who's had a diagnosis of mild cognitive impairment and at least one biomarker to confirm that underlying pathology. And so of late, we've had some lovely breakthroughs in the field more broadly, which have come through immunotherapy. And so a couple of antibodies, monoclonal antibodies, have recently demonstrated in the gold standard of clinical trials, in phase 3 clinical trials, that they've been able to reduce the amyloid plaque burden in the brains of patients suffering from Alzheimer's or prodromal Alzheimer's. And that reduction in amyloid plaque has correlated with a slowing of disease progression. And there's an important point there, that slowing of progression is not, we're not yet talking about cure of disease or even prevention of disease. So the patients who've been treated with the monoclonal antibodies have had roughly a 30 to 35% slowing of disease progression. That's the first success this field has enjoyed in many decades of research. And so as a first step, it opens the way for subsequent therapies to come in and hopefully have a positive effect. But equally important is this large segment of the pie, what we're calling preclinical Alzheimer's disease. And I think the aim for any new treatment, if it is not curing an existing condition, is the prevention of such a condition. And particularly neurodegeneration, where you end up having the loss of nerve cells and neurons. what comes with that loss of neurons is a loss of function, loss of memory, loss of ability to conduct daily living activities. And so the ideal approach to managing Alzheimer's would be to stop it before it begins in the first place. You preserve quality of life, preserve function, preserve independence for much longer. And while the recent successes in these more advanced patients are very good and promise an avenue to the future, those particular monoclonal antibody-based treatments are probably not appropriate for this very large population of over 300 million people who are at risk of developing Alzheimer's disease. So the total picture here, 400 plus million people suffering from Alzheimer's disease. If we could one day imagine a way to manage the earlier stages of diseases, we might then ultimately be able to talk about prevention. And some of the approaches that we can talk about today are vaccination-based approaches, and they offer a number of attractions relative to these monoclonal antibodies. While at heart, the approach is immunotherapy, the vaccination approach has a number of advantages, which I'll come to in a moment. And so the path towards dementia has been mapped quite accurately of late, and there are a number of stages which can be identified on the basis of molecular pathology, which give us a window of opportunity in terms of treating patients at earlier stages of the disease evolution. So someone with Alzheimer's will have brain accumulations of aberrant proteins, amyloid, tau, TDP-43, a variety of others, alpha-synuclein. So this patient at the end of this course of pathology is probably beyond helping in any significant way. What we want to do is target therapies to patients who have mild cognitive impairment, or even those who have preclinical AD, or better still, no symptoms at all. But we can't just treat every elderly individual in the population. That would be impractical, probably not ethical either. So we have to be able to identify individuals who are at risk and who can then benefit from some of these potential interventions. And so on the left here, you see a scale where disease develops over time, where you begin with relatively limited pathology, and over the course of 15 to 20 years, you see changes in the molecular characteristics of certain proteins in the brains of these individuals. And one of the first to occur is a change in the amyloid beta protein, which you see increasing quite early in the disease process. And this can be mapped, also correlates quite nicely with amyloid PET imaging. So positron emission tomography is an imaging utility that you can use where you are able to track the occurrence of these aberrant protein aggregates quite nicely using specific traces. And there are other traces that can come along, and tau is one that particularly correlates very well with the accumulation of disability in neurodegeneration. So there are a number of now molecules that we can use to track the increasing risk and the development of pathology in individuals, but at a very early stage. So if we were able to start to introduce therapeutic interventions at this sort of stage, we then have the possibility to impact quality of life, preserve neuronal function, and so on. And so ultimately the aim is to start to intervene at this point in the disease development. But to do that, we must have treatments which are safe, obviously effective, but the safety is a critical element of this story. So at AC Immune and many other companies, we're now looking at the challenges confronting us in neurodegenerative diseases and the dementias. And one of those is the fact that With Alzheimer's disease, it is not simply a pathology of amyloid beta. There are a variety of other proteins which are hallmarks of neurodegenerative diseases, and many of these can actually play a role in Alzheimer's disease as co-pathologies. So A beta is clearly a dominant protein and is thought to be a key trigger for the development of Alzheimer's disease. But tau is also very often a co-pathology. And then we see some of these other markers of disease in the brain, alpha-synuclein aggregation, particularly well associated with Parkinson's disease and Lewy body dementia, but it is also present as a co-pathology in many patients with Alzheimer's disease. So with this constellation of proteinopathies in many of these neurodegenerative diseases, it's really important that when we are talking about therapy for patients, we're trying to target the right sort of therapy to the right patient. So that therapy may be an amyloid beta targeted therapy. It may subsequently then be followed by a tau targeted therapy and so on. So we're trying to match the pathology that people experience with the possible treatments that are now still in development. And one other thing, I mentioned that we have these co-pathologies in the antibodies which recently showed success against Alzheimer's disease. So that's lecanemab and donanemab that have shown that success. These antibodies don't have a uniform effect on every patient that's been involved in the trials. So there are some patients who respond very well to an A beta monoclonal and others who don't respond at all. And it may well be that the different types of co-pathologies that exist in the different patients may be impacting the level of response to the A beta antibodies. And so to get closer to this long-term prevention type of treatment of Alzheimer's disease, but not just Alzheimer's, we may also be talking about Parkinson's or ALS or a number of other conditions. What's so intriguing for us, at least, is the way that an immunotherapy has now demonstrated a success. And so by immunotherapy, we are mostly thinking about therapies with using antibodies. Antibodies do penetrate the blood-brain barrier, and they are present in the extracellular space and the CSF. So antibodies have an opportunity to interact with target structures, the aberrant proteins that are associated with disease. So you can see here we've got an amyloid plaque that is composed of, it looks fairly random, but it is actually composed of highly ordered structures, which are these fibrils of A beta, and ultimately each fibril is composed of a series of oligomers, monomers of A beta that misfold, and that misfolding acts as a template for other elements to then misfold on top of them. So you get a very ordered structure, but it is an aberrant misfolded protein at the origin of that structure. So if you have an antibody which could interact specifically with that disease-related protein, the misshaped, misfolded protein, then you have a way of preventing the pathology from extending throughout the brain. And this would be particularly relevant in the case of tau and amyloid beta. So the idea, get an antibody into the brain where it can block the function of these aberrant proteins, and in so doing also lead to the removal of these proteins. So when an antibody binds to a target antigen like this, there's a change that occurs in the tail section of the antibody, which makes it a good target for uptake and degradation by antigen processing cells. So in the brain, these are the microglia, and we know these cells have the ability to remove the antibody conjugated or binding to the target structure. So ultimately, we hope to remove these aberrant structures using the antibody and the microglia to lead to what would hopefully then be a healthy neuron. And so I spoke recently about the success of the monoclonal antibodies. In the sense, these have begun a process where the field can now move towards a greater understanding of the pathology and how we might be able to interfere with that. So in each case, in the case of lecanemab, this is an antibody that reacts with oligomeric amyloid beta, and donanemab, which reacts with a pyroglutamate version of amyloid beta as well. Both of these monoclonal antibodies, when administered, in the case of lecanemab, it's administered by infusion every 2 weeks, and in the case of donanemab, it's an infusion every 4 weeks. Very soon after the commencement of therapy, you're able to detect a lowering in the amyloid plaque level in the brains of the patients on the study. And so it happens very quickly. Within 3 months, you can already see a statistically significant change. By 12 months, most of the patients have got levels of amyloid plaque which are low or undetectable almost. And that removal of amyloid plaque in both cases has been correlated with a clear, maybe modest, but a clear reduction in the rate of progression of when you're looking at a functional scale of cognition and daily living performance. The patients who are being treated with lecanemab or donanemab are seeing a benefit of some 30% or so in terms of slowing of the rate of progression of the disease. So clearly there's still a lot to do in making sure that or trying to halt disease progression completely. And if we want to extend that further to prevention, there's a long way to go. But the underlying mechanism of action of these antibodies, as I showed on the previous slide, is to interfere with the amyloid beta aggregation and the pathology caused by that. And so we can see that removing plaques correlates very nicely with the reduction in disease progression. There was one other point that I wanted to make here, but it escapes me. Perhaps I'm already starting to suffer from some of these memory problems. But in any case, we've got a mechanism of action which gives us a good sense that we can do the right sort of things at intervention. A last and important point about these trials is that the patients who were enrolled in the studies were characterized as having a mild cognitive impairment and already some clear markers of underlying pathology, notably amyloid PET positivity at a certain level. And the fact that those patients were entered into the study with a baseline imaging program is important, because in many of the earlier studies of other antibodies, also directed against A beta, many of the patients who entered the study didn't actually have amyloid pathology. And so we now have the way to identify patients as they're coming into the study to make sure they have the underlying pathology that we are targeting, and then we have ways of evaluating the changes in that underlying pathology over time. And so the field of vaccination benefits greatly from the work on these antibodies, because for a long time, the whole amyloid hypothesis in Alzheimer's disease, there was a lot of debate about whether it was actually relevant to the development of disease and a relevant target for therapeutic intervention. And so with these immunotherapy approaches, monoclonal antibodies, we know that you can target the beta as an example, and interfere with how the underlying pathology develops. And that's using an antibody-based mechanism of action. And so by trying to replicate that passively administered antibody therapy, we're trying now to use an active immunotherapy, what we call, or vaccination. So there are a number of vaccines in development across the space. There is our own A beta vaccine that we call ACI 24.060. That's quite a mouthful. And there are a number of other companies developing vaccines similarly targeting A beta. The field comes with some baggage in terms of the vaccines that have been developed for Alzheimer's, and the very early candidates, and we're talking about 2000, 2001, there were a couple of candidates that were entered into clinical development, and they actually caused some serious side effects. And that was partly because they included the full-length amyloid beta protein. So that's a protein that has between 40 and 42 amino acids. And in that sequence of amino acids, there are a number of epitopes for T cell reactivity. And one thing that was learned as a result of those developments was that target-specific T cell reactivity in the brains of Alzheimer's patients was not a good thing. It caused side effects, some quite serious, and there were even a couple of fatalities in the study. So that really stopped the development of the vaccine programs for some time, whilst the understanding behind the toxicity was increased. So today, most of these vaccines do not encode or do not include the full-length sequence of the amyloid beta protein. There is one exception here, but the way the protein, the peptide, is presented limits the amount of T cell reactivity that's induced. In our own case, we've chosen the first 1 to 15 amino acids in the N-terminal part of the amyloid beta sequence. There is one vaccine that just a couple of days ago at the CTAD conference in Boston produced some results in a phase 2 study. They are using the C-terminal 7 or 8 amino acids of A beta, and then there are a couple of others who are looking at the N-terminal end, and one which is looking at a quite different construct. But this last one is also looking at a patient-derived, so a very highly bespoke product for patients where their own dendritic cells are pulsed with this full-length sequence. One of the other things too, so each of the vaccines comes with a slightly different structure. Our own, which I'll come to in a little while, is a liposome-based vaccine. There is this hexameric conformation version. There is one which is conjugated to KLH. That stands for keyhole limpet hemocyanin. There is a challenge behind this particular component of the vaccine in that most of the pharmaceutical regulators, like the FDA or EMA, will really have a difficult time approving something like a KLH-based conjugated vaccine, because this KLH is itself a very heterogeneous product extracted from keyhole limpets, which is not easy to come by. A DNA vaccine has its advantages, certainly in the synthetic side of developing vaccines. DNA is easy to make, and as we've all noted with the COVID vaccines, the mRNA vaccines, something based on a nucleic acid you can produce at mass and quite cost effectively. There is a challenge I see with this particular approach in that you are not focusing necessarily on the pathologic species of A beta, which I will come to again. And finally, then there's another conjugate-based vaccine that has also been in phase 2 development. If you're not aware, a lot of vaccines come these days with adjuvants. These are like an accelerator for the immune system. In our own case, it's monophospholipid A. This is an element that's used in many vaccines for infectious disease and is well known to the regulators. Alum is, again, another commonly used component in many vaccines. And finally, then CpG, this is a motif of DNA that's often found in infectious agents that is also good at accelerating or exciting the immune system. The adjuvants have an important role in enabling the immune system to be presented with the right sort of structure. And finally, then, there are other elements in many of these vaccines, which we're calling T helper cell epitopes. T help is an important part of most natural immune responses and immune responses to vaccines. It ensures that the B cells, the antibody producing cells of the immune system receive the right sort of accessory signals for the immune response to evolve appropriately. And so many of us are including T helper cell epitopes, but these are not epitopes that are related to the target antigen A beta. These are mostly things that come through natural exposure to different infectious agents, influenza, hepatitis, or through childhood vaccination programs, things like tetanus toxoid or diphtheria toxoid. So the T cell help ensures that you get a long-lasting and effective antibody response. And so on to our own example of a vaccine against A beta. We have a platform at AC Immune which we call supra antigen. And basically, this is a liposomal formulation where we have the target antigen linked into the membrane of the liposome, chemically linked. And the reason for that, I alluded to it earlier on, is we want this antigen to look like the pathologic version of the protein present in disease. And so this cross-linking of the antigen into the membrane of the liposome makes sure that the target antigen adopts a beta-pleated sheet structure. And so many of the misfolded proteins, when they aggregate in this highly ordered fashion, are actually presented as beta-pleated sheet structures. And so by doing this palmitic chain anchoring of the target antigen into the surface of the liposome, we are presenting to the immune system a structure which looks like the pathologic A beta. And as well as that, we have these adjuvants, as I mentioned, MPLA or CPG, and then a variety of T cell non-target, so non-A beta related T cell helper epitopes. So all of this combined gives you a target-specific antibody response, and by that we mean an antibody response targeting the pathologic species of the protein, be it A beta or phospho-tau or one of the others. And then we get the right level of T cell helper activity to ensure that the antibody response we get at the end ticks all of these boxes. So the protein target is being well recognized in an immunogenic form. You get lots of antibodies that are specific for that pathologic form and not for the healthy form of the protein. And that the antibodies, over time, they increase their affinity for the target structure, and they can be reboosted with subsequent immunizations. So this, we believe, is a highly effective platform. We have some clinical data with a vaccine that we have in collaboration with one of our pharmaceutical partners that has shown really excellent antibody responses, reacting with the pathologic form, not the normal version of the protein. We're confident that this structure can deliver the sort of response we need to see for a long-term prevention approach. And so the first vaccine that we have taken into the clinic is one we're calling ACI-35. The target antigen is phospho-tau. So tau is a normal protein that's present in the brain and actually throughout the nervous system. And when it's phosphorylated, it then begins to exhibit this propensity to form these unusual aggregates that can be quite toxic to the cell. So this vaccine, you see our supraantigen-based version of it, was tested in a head-to-head study with a candidate, which was a conjugate vaccine called called JASI. So we've got our ACI, and JASI was the in-house candidate from Janssen Pharmaceuticals, which is a division of Johnson & Johnson. And so testing these two in a head-to-head study showed that both vaccines actually give you a relatively good antibody response against the pathologic species of tau. So EPHF is enriched paired helical filaments. These are the larger aggregates that you get with tau when it misfolds. So developing a good level of antibodies against that is key to the puzzle. One thing of difference between the two vaccine candidates. So the JASI vaccine required 2 shots. So the immunizations are shown along here. It required 2 immunizations before it started to induce a reasonable level of antibody reactivity against EPHF. Whereas in contrast, the ACI-35 vaccine, 100% of patients in the study developed antibodies against EPHF after the first injection. So you're saving a couple of months of time in terms of developing an immune response against the target antigen structure when using ACI-35 compared to JASI. So that was a small win for ACI-35. But then when you're looking at the antibody response against normal, non-pathologic tau, you see a difference in the antibody response pattern. We were relieved in some way to see this difference emerge. So JASI induced a high level of antibody reactivity against tau, whereas ACI-35, it saw a spike at the beginning, but then that antibody response diminished over time to almost baseline levels. And it's this mixed profile, which is really what you want to see when you're taking a vaccine into development against a protein which is actually part of our normal healthy makeup. So tau, as I said, and A beta alike, these are normal proteins fulfilling normal functions. And in the disease state, there is a pathologic form of those. So you want your antibody response to distinguish very sensitively between the response against the pathologic species and the healthy species. So we have a perfect profile here in some ways, where you have limited reactivity with the healthy protein and good reactivity with the disease protein that comes on rapidly. If you imagine using these vaccines in a long-term setting, if you have this sort of reactivity, a high level of reactivity against the normal protein, you could imagine that it may lead to tolerability issues and toxicities over a long period of time. The other final important point that I'd like to make is that all of the individuals entering this particular clinical trial were elderly. So I think the age was 60 years or so of age. And I'm sad to say, given that I'm in that age bracket, I'm sad to say that the immune system in such individuals is not always as able to respond as effectively to immunization or immunological trigger. And so seeing 100% of patients respond with the first injection, it says something about the incorporation of adjuvants in the vaccine and those T-cell helper epitopes. So this is a good vaccine profile to take forward into an elderly population, potentially also ultimately in a prevention setting. So that's vaccine number 1 in our portfolio. And I'll just spend a couple of moments on ACI-24, which is our A beta vaccine, where we given the results that have happened with lecanemab and donanemab, we are now encouraged to take the A beta vaccine forward into development. And we have started a phase 1b/2 clinical trial last year. But before that, we have some data from non-human primates, which encourages us tremendously as to how this vaccine might function in man. And so the first panel here is the level of antibody response against oligomeric A beta. So we see with vaccination, we get a very good level, boostable and statistically significant level of antibody against oligomeric A beta. That is important because the the monoclonal antibody lecanemab reacts with that same target structure of A beta. So with our vaccine, we're inducing lecanemab-like reactivity against oligomeric A beta. Additionally, our vaccine is inducing antibodies against pyroglutamate A beta. And this particular structure has been known to be highly inducive of amyloid plaques. It is often found at the cause of the plaques. And so our immunization is inducing antibodies at a good level against pyroglutamate. Pyroglutamate is the target for this other monoclonal antibody, donanemab, which, again, recently in phase 3 studies demonstrated very good success in terms of removing amyloid plaque and having that benefit of slowing disease progression. So we're happy with the antibody profile and the antibody specificity that's coming with ACI-24 vaccination. And when we look at the scope of reactivity of the antibodies against A beta peptide, the full length A beta peptide or fragments of that, we see that the vaccine really is targeting antibodies against the central portion of the 1 to 15. So that's actually 3 to 15 amino acids in the N-terminal sequence, which is the ideal, and we're not seeing much reactivity outside of the immunizing target structure. So we're getting a nicely specific response against the immunizing target structure, and that is replicating the activity of now 2 monoclonal antibodies that have demonstrated the desired activity in phase 3 clinical setting. So our antibodies in these animal models have been shown. So this is data now from a double transgenic mouse model, which is it develops a quite aggressive amyloid disease and at a very early age. So in the control animals, you see the development of amyloid plaque on histology, quite a heavy load of amyloid plaque. In the animals that were immunized 3 times over the space of 6 weeks with our ACI-24 vaccine, you see that's greatly reduced. And in terms of the numeric effect, we're seeing roughly a 50% drop in the area of amyloid plaque in the brains of these animals and in the number of plaques. So this sort of observation, plus the observation of strong levels of antibodies against oligomeric and pyroglutamate A beta, give us confidence of taking this vaccine into the clinic as we've done. So it's now in a phase 1b/2 study in both sporadic Alzheimer's disease and a population of individuals with Down syndrome. As a side, I'll just talk about this Down syndrome for a second. Individuals with Down syndrome have the extra copy of chromosome 21, and coincidentally, unfortunately, that also includes an extra copy of the amyloid precursor protein gene. And so from an early age, individuals with Down syndrome exhibit high levels of amyloid protein in the brain, and from about the age of 40, begin to develop Alzheimer's-like disease. And so this is a population that is usually not included in trials of amyloid targeting therapies. And we've found in collaborating with them that they're extremely motivated to participate in the studies, both the individuals themselves and their carers. But there are other issues in doing clinical trials with such population. So this is something that happens in the development of ACI-24 under very strict supervision. The Alzheimer's, the sporadic Alzheimer's disease, this is the 400 million population. We have already completed recruitment of 2 cohorts. The second of those was expanded, and we're beginning a 3rd cohort as well. One of the attractions of this particular study is that it's adaptively designed, which means that we can do numerous interim analyses to modify the trial if needed, and we can look at the data as it emerges in a randomized, this is a randomized study, but we can look at the data in a blinded fashion without any penalties there. So we're able to look at the safety signals as they're evolving, but we can also look at the immunogenicity, so the levels of antibodies that are being induced in the different cohorts. So that trial is progressing well. We will have further data on the sporadic Alzheimer's disease population by the end of the year, so more safety and immunogenicity data. And then most importantly for this study, and because of what's happened with lecanemab and donanemab, and the correlation now that's quite very well accepted regarding the measurement of amyloid PET and how that correlates with cognition, we will be reporting the first amyloid PET data in this study for patients who've received the vaccine for at least 6 months. So we're quite excited to see that data. We're also quite nervous, I'll be honest, and the reason for that is that when you give a monoclonal antibody, you're giving a bolus of active agent in a single sitting, and then you're topping that up again 2 weeks or 4 weeks later, and so the kinetics of that response could be quite quick. Whereas when you're immunizing with a vaccine, looking to develop antibodies against what is ultimately a self protein, it may take a little bit longer for that for those antibodies to reach a desirable therapeutic level. And so we may need to wait a little bit longer than 6 months. We will be looking again at 12 months for sure. In any case, we're excited to see this data emerging when it does. And so I'll end on a couple of general points, if I may, and that is that that vaccines targeting these aberrant proteins that are occurring in neurodegenerative diseases, they make a lot of sense and they hold a lot of attractions in the therapeutic setting. And one of those is that with a vaccine, you're encouraging the body to develop its own antibodies against the target antigen. And so instead of relying on an external manufacturing process to make antibodies of a single type, here we're producing a polyclonal antibody mix, and that immune response can be quite long-lived, and it delivers a level of antibody which remains at a good level for a reasonable period of time, many months in some cases, and that can be reboosted at periodic intervals. If you compare that to the monoclonal antibodies where you require dosing every couple of weeks or every 4 weeks, there's a very different burden of compliance in those two settings, and we foresee ultimately that after a first priming year with a vaccine, you may move to a situation where you only need a vaccination every 6 months or every 12 months. So that dosing regime, the consistency of the effect are obviously quite beneficial in the vaccine setting. Important as well is that in our vaccine studies to date, we have not seen any of these potential side effects, which we're being called ARIA. That stands for amyloid-related imaging abnormalities. And you have either edema, so brain swelling, or microhemorrhages. But in both cases, we've seen no ARIA-E, and certainly with the monoclonal antibodies, they're reporting an incidence of somewhere in the range of about 10% of patients developing ARIA-E, which is of concern. It requires close monitoring, sometimes discontinuation of treatment, and that monitoring also includes brain imaging, and that's not necessarily a straightforward thing to imagine when you're talking about patients who are suffering from dementia, and the anxieties associated with imaging. Not great. Finally, then there are certain economic and logistic advantages with vaccines. vaccines, they can be stored on a shelf. In most cases, not all. In the case of mRNA vaccines, that's not necessarily true, but in the case of what we're looking at here, we could probably have these shelves stored for at least a year. I think we're up to 2 years at the moment with our own formulation. So the economics and the infrastructure requirements for administering a vaccine, they're much more favourable than where you might need an infusion centre, and actually the cost of some of the monoclonal antibodies is very high. I think I could live with that. There are a few more slides in this which are much more corporate in terms of AC Immune, but I think for the moment I'm happy to stop there and we could begin the discussion. Or Maria Teresa, if you're able, you can lead the conversation.
Sure. We're ready. Can you hear me?
Very faintly.
Okay, great. Okay, so actually, this was a great presentation. So you provided a very updated overview of what is going on. So there are several questions now on the chat, and one is okay, I lost some. Okay, so let's start from. Okay, the first one says, how do the mechanisms of a potential Alzheimer's vaccine differ from traditional vaccines, and what unique challenges are associated with developing vaccines for neurodegenerative diseases?
Yeah, it's a good question, and so typically when we think about vaccines, we think about infectious agents, and in a previous life we also imagined vaccines for cancer. So vaccines for infectious agents, COVID is a good example. You have a defined risk, and you have target for the vaccine which is not part of our own biology. So a coronavirus or a human papillomavirus in the case of cancer, these are structures which we can relatively safely develop an immune response against. And there is little in our immunological history that would suggest we cannot respond to those infectious agents. The contrast with neurodegenerative disease is that we typically, and maybe someone can correct me, we typically do not have an infectious agent as the trigger of disease. And so the disease pathology itself is related to our normal proteins, A beta, tau, alpha-synuclein, and so on, where they fulfill a normal function, and our immune system, which we call tolerance in immunology, and basically that is the process when the immune system develops. It is trained not to recognize ourselves, and so we eliminate, as we're developing in utero and in the years that follow that, we eliminate self-reactive components of the immune system, and that process, tolerance, makes it actually very hard to generate immune responses against self-proteins. Now the system does go awry occasionally, and we have autoimmune diseases like rheumatoid arthritis and a variety of others, where you get you see the toxic nature of the immune system if it reacts with our self components. So by vaccinating to manage the development of a neurodegenerative disease, we want to make the immune system firstly see something which is part of our normal biology. We want to target that response, as I mentioned, we want to target the response against the pathological determinant. So it's this misfolded version of the healthy protein that we're trying to address. And that's not easy. That's why we incorporate the adjuvants in the vaccine. It's why we need the T-cell help. And so, we're using all the tricks of the trade, as it were, to ensure that we can generate antibodies against those pathologic species. And in fact, some of the vaccines, and as I showed on the ACO35 trial, The JASI vaccine has a conjugating component, a conjugating protein with the antigenic structure stuck out on the side, and the antibodies that reacted with that antigenic structure actually reacted with the normal tau as well. So, we have to be careful about what we induce the immune system to see. The antibodies that we generate must be specific for that pathologic species. Otherwise, we risk all sorts of complications that we'd rather not have in a long-term therapy. Did that answer the question adequately?
Yes, thank you. So I lost connection, so I cannot see the questions anymore, but I remember some. So the next one was on the accessibility. So in the next future, what will be the rules for the enrollment of people to the access to the vaccines? Yeah.
So accessibility is one of the things that we are actually quite concerned about, and we want to ensure that the greatest possible population does have access to such treatments. And in the case of monoclonal antibodies, which today are the only disease-modifying therapeutic options available, not yet everywhere, the antibodies are extremely expensive and not easily administered. Yes, if you live in the US or in Europe, anywhere, you can infuse an antibody over a period of 2 or 3 hours. It's not complicated, but it requires infrastructure and it requires cold chain storage and a whole bunch of things which are a challenge. But most of the challenge comes in the cost of these therapies. So some of the antibodies are costing $25,000 for the course of treatment, and that's beyond the means of most people in the world, actually. And so the 400 million people that I spoke about in one of the earlier slides includes probably the majority which are not based in the US or Europe. And so we believe the vaccine approach, because you're not requiring a complicated pharmaceutical chain for manufacturing massive amounts of monoclonal antibody, we believe the vaccine approach offers the attraction that it can be cost effective. And so if we can transmit a vaccine around the world, the logistics are relatively straightforward. If the vaccine does not require cold chain in the supply network, then it's also, again, easier to distribute that, and certainly at a cost far below the $25,000 that might be involved with a monoclonal antibody. Then on the side of ensuring that we get most relevant population coming into the clinical trials, that is actually more complicated still. When we're developing vaccines, we need to make sure that the regulators are going to be satisfied with the quality of the study that's done, the quality of the way the data is held and manipulated, and so on. So we are limited to an extent in terms of doing the initial testing for the vaccines in Europe and the US, or North America, let me say, and some of the other Western style economies, and with data emerging from those centres in a sort of a quality controlled manner, then we hope to ultimately, step number one is get the vaccine approved, and from that point on, we will then look at the accessibility issues on a wider basis.
We can proceed to the next question in the meantime. So, are there ways to know some subjects may develop unknown side effects before actual vaccination?
Good question. I have to say I don't think so, unfortunately. What we're trying to do when developing vaccines is to ensure that the people who come into the study have the appropriate pathology, there's no point raising an immune response in someone if they don't have the underlying pathology. That's a kind of a waste of their immune system. So we try to ensure that with a positive mindset that we are in the right patient. Now there are other things that would make us exclude patients from studies, so not having the pathology is point number one. We would typically prevent people from entering a vaccination study if there are known sensitivities to any of the components that are included in the vaccines, and if the subjects are exhibiting or have other comorbidities that would give us cause for concern about their ability to safely generate an immune response. So there's no single determinant that you could identify in someone that would put them at greater risk, other than perhaps sensitivity to any of the vaccine components. But as I mentioned, the vaccine, in our own case, the components in that vaccine have mostly been used in other vaccines, so that the adjuvants and the T-cell helper epitopes are well known to most of us in our immune systems, and also then to the regulators who would determine whether we can take a product into human clinical development or not.
Okay, thank you.
Yeah. Okay, sorry, Léa, can you hear me?
Yes.
Very well.
Yeah, now I got the questions from email, so perhaps now I will succeed. So
Okay.
the next one is, do the treatment affect neurobiological mechanisms inducing the accumulation of amyloidosis and tauopathy and the production of the circulating species? You got it?
Yeah, I got that question. I've also now got an echo, but that's good. Do the treatments so in our own case, I can't say yes or no. It's simply too early to report, and we haven't got that data. We will be looking at all of the sort of the battery of clinical tests to evaluate patient performance, cognition, memory, daily living activities, and so on. As I've also mentioned, we're looking at the amyloid plaques themselves, and in some of the trials where they're also looking at tau PET. So the effect of the treatment is being evaluated as far as the disease behavior is concerned, and I think that's a fairly gross level of evaluation and examination. We're not looking in any more sort of fine level of neurological function other than what is exhibited as clinical symptoms for patients. Is that answering the question? No, I don't hear you again.
Oh, sorry. Yeah. Somehow, yes, it answered the question. There is, of course, more to do, but still, we have some hints, apparently.
So while we're using immunotherapy for some of these trials, and the monoclonals and our vaccines, we have to accept that immunotherapy is a valid mechanism to target some of these pathologies only at certain stages of the pathology. So tau, for an example, part of the early stage of tau pathology is an intracellular misfolding of the protein, and so an immunotherapy won't work in that stage. It will only work when the misfolded protein enters an extracellular space. Antibodies don't get into cells effectively. So when tau pathology spreads through the brain, then at that point an immunological mechanism would be effective. But in the earlier stages, we believe a small molecule approach is more appropriate. And it's for that reason that AC Immune has a small molecule platform, and we're developing tau aggregation inhibitors, we're developing aggregation inhibitors for alpha-synuclein and so on. So, we need to think of the ultimate approach to any of these neurodegeneration therapeutics. Might require combination approaches, it might require sequential approaches. So target A beta early first to limit the amount of tau that is triggered to misfold, and maybe use a small molecule early as well. So we try to ultimately imagine a day where we would mix and match different mechanisms of therapeutic intervention, antibody-based through vaccines or small molecule-based.
Okay, so ready for the next one. Okay, what do you envision as the most significant challenges in rolling out an Alzheimer's vaccine on a large scale, and what strategies are in place to address these challenges? Thank you. Tough questions.
Yeah, and not easy to answer that one. I think step one for us is going to be get the vaccine, or demonstrate the vaccine is safe and effective in the sort of robust clinical trial setting that we all know. Following that, rolling out a vaccine across the world, we know it's possible. We know it's possible in a very short period of time, and the COVID experience has taught us many lessons about how to do that. For the moment, we are, it may be premature to think about distributing an Alzheimer's vaccine around the world today, because we are still many years away from having an approved product. But despite that, we already have engaged with a number of the international organizations to begin thinking about how it might work. We know if you have a vaccine which can be cost effectively manufactured, does not require a complicated cold chain in the supply chain, then you can actually deliver it quite effectively internationally from the top to the bottom of the globe. So I would hope that if we get to the point where we can do that on a logistic level with an approved product, of course, then that would be great. One of the challenges that we haven't really spoken about, and that is this comes back to our thinking about precision medicine. And so we would simply not give this sort of vaccine to every person over the age of 50 or 40 or whatever the cutoff you might think. You simply would not do that, because that wouldn't represent an effective use of resources, and not everyone is going to develop a neurodegenerative disease. So we would really want to restrict use of such a product to those people who are at risk of developing the disease. And I mentioned early on there are certain biomarkers for Alzheimer's disease, and there are blood tests which are coming, I think, around the corner. The Swedish groups are doing some great work in looking at constellations of different blood biomarkers. And so you can imagine that you could ultimately identify individuals at sufficient risk of developing disease, where you could then imagine using the vaccine based on a blood test. If someone has a particular constellation of P217 tau and A beta ratio, then you could imagine suggesting using a vaccine in such individuals. But simply doing tests like that is not necessarily straightforward. If it could be done by an ELISA, then yeah, you could imagine it could be rolled out worldwide. But if it requires high throughput mass spectrometry, you might have to think otherwise about how applicable it would be in some developing countries.
I was asking, what about genetics to identify people that have an high risk?
So we did a study with one of our programs, not one of the vaccines, but one of the monoclonal antibodies that we were developing at AC Immune in collaboration with Prof. there was a study done over a period of 9 years. I think it began in 2013 or 2014, and it was done with a family cohort based in Colombia, and all of the individuals enrolled in the study had a mutation in PSEN1, presenilin-1 gene, and it's if you have the mutation, the chances are probably 99% that you will develop a precocious early form of Alzheimer's. Usually from about the age of 40 to 45, you start to exhibit those symptoms. And so the study of this antibody was done where and it was the family cohort numbers many thousands of individuals. The study enrolled, I think it was something like 200 individuals, all confirmed with the mutation, and ultimately the study was partially successful, let me put it that way. It did not hit the primary endpoints, but the idea was that you would give the antibody, this is an anti-A beta antibody, and try to prevent the development of pathology by inhibiting the formation of the plaques. And the antibody itself was known to bind to the oligomeric form of A beta, but I think the way the study was done, it led to complications. And of course, over a period of 9 years, when you start a study, the 9 following years, you're learning a lot more about the disease from the field. So there are things in the way the study was done which were probably not optimal. One of those was enrolling patients, not patients, sorry, they were individuals with the mutation, but they were enrolled probably at a 5 years too young age. So the first 5 years of treatment in the study were providing an antibody to individuals who had no development of pathology in that period of time, in both the treated and the control cohort. And so the real therapeutic part of the study only was the last 4 years. And in that time period, we had a bit of COVID interfering with how the study was done. And the level of antibody that was administered was also adjusted. In the end, the study was, even though it was 9 years long, it was probably 5 years too short because the individuals at entry were too young. And so I think that there are useful, thinking about pathology and the way it develops, there are useful genetic markers of disease. PSEN1 is one of those. Excuse me, I mentioned the Down syndrome cohort that's being done in our A beta vaccine. The additional copy of the amyloid precursor gene is known to lead to high levels of amyloid protein in the brains and subsequently the development of Alzheimer's-like disease in Down syndrome. The challenge in looking at genetic markers of the disease and how it's if they're disease related is actually running a study in enough individuals. So I think the other genetic markers of disease, there might be something like 4 or 5% of individuals who develop Alzheimer's have a genetic link somewhere in their makeup that is considered related or causative of Alzheimer's. So doing studies in genetic Alzheimer's is actually a real challenge unless you have those wonderful cohorts of nature, like in Colombia or the other populations that I mentioned.
Okay, so Gary, there is a long queue of questions.
I'll try to answer more shortly.
Okay, so the next one is about how, if it is possible to translate the knowledge on Alzheimer's vaccine to prion disease.
So we now think of these neurodegenerative diseases, A beta, tau, alpha-synuclein, TDP-43. There's probably a couple of others to add in there. we think of them as prion-like diseases. And so you have the same kind of proteinopathy, as we're calling them. So yeah, they're fascinating from a molecular disease perspective, but actually trying to treat them is a real challenge. Normal proteins fulfilling normal functions, for them to suddenly become abnormal and misfold, and that misfolded protein then acts as a template for other elements of the species or numbers of the species to similarly misfold and aggregate and cause pathology. It's a fascinating way of thinking of protein disease as being infectious. Are we going to take this approach into the treatment of prion diseases? I think that's an academic research program, and there is definitely no disrespect meant in that. It's simply not possible for a company doing this very expensive biotech or pharmaceutical development to do such a thing. I can't imagine where to start. Actually, it would be just a challenge.
Also because it is a quite rare disease, so it's not worthwhile for a company, perhaps. Yeah.
Perhaps.
So academic is better. So let me say to the audience that I am really amazed because questions from the audience, they are brilliant questions, actually. Okay, so the next one comes now. Okay, so how will personalized medicine and vaccine strategies adapt to the heterogeneity in Alzheimer's disease presentation and progression, considering specific biomarkers of patient subgroups and that show potential in this context?
That's a good question, and one of the reasons why I showed that plot where we have 5 candidate targets. Let me say, so A beta, tau, alpha-synuclein, TDP43, and NLRP3 ASC. So it's fascinating when we look at the responses that lecanemab and donanemab are inducing in treatment. We see that not all patients, it's not every patient benefits 35% slowing of disease progression. There are some who are non-responders, some who are good responders. And we do wonder whether the co-pathologies dictate how much a patient might be able to respond to a single agent therapy. So A beta, a single agent therapy, going into someone with A beta only as the pathology may be very effective. If you have that patient also having a TDP43opathy, and actually we're probably looking at 60 or 70% of patients with Alzheimer's might also have TDP43 aggregates. And so a co-pathology like that may limit the amount of the effectiveness of your A beta targeting. And you may also have to then consider dual targeting with both A beta and TDP43, or perhaps in that individual going earlier. It's actually, I think one of the biggest challenges in neurodegeneration is accurately mapping the underlying pathology. And it kind of we could think of cancer years ago, and in the 90s, not everyone with lung cancer, everyone with lung cancer was thought to have the same disease, and we know now there are different. Same for colon cancer. There are some who have some genetic markers of the disease and will respond to some drugs, and others not at all. So we're getting to that point, I think, in neurodegenerative disease where we need to really understand what's going on underlying the clinical signs of disease.
Okay, then the next one, it seems to me that you might have already answered within your presentation, but by the way, so the question is, are there different types of Alzheimer's disease vaccine under investigation by your company, and what are their unique approaches and potential advantages?
Okay, so we do have multiple vaccines in development at AC Immune. One targets A beta, one targets phosphorylated tau. I showed the data from both of those. We have a third vaccine in development targeting alpha-synuclein. That vaccine is actually being tested in a study for Parkinson's disease. But we could well imagine that if it proves to be efficacious and safe, that it could also be used in patients with other dementias where alpha-synucleinopathy is a feature of the underlying pathology. So we have these vaccines against individual protein pathologies. They are being developed for clinical indications like Alzheimer's or Parkinson's, but you could imagine if they demonstrate efficacy against the target antigen, that they could be used in any clinical setting, actually, assuming that the patient has the underlying pathology.
What efforts would be done to encourage people to be vaccinated earlier in life, considering the late onset condition of Alzheimer's disease?
Yeah, good question. You can speak from experience if a family member has had disease, or if you consider yourself at risk because you've done a test for ApoE4, for example. So there's one genetic marker, ApoE4, that there's a raised risk of developing Alzheimer's. This is actually a tricky one, because there are parts of the population who have a natural hesitancy towards vaccines. I'm not one of them, but I would say if you have a safe option to potentially even prevent a disease, it's worth undertaking or using that option wherever possible. I have it is a challenge when you think of genetic tests for certain risk profiles for diseases, and some people just simply don't want to know the result, and because there's no therapeutic option. And I can understand that in those sorts of cases, but where we might have a suitable, safe alternative that could prevent a neurodegenerative disease, and some of the diseases are really terrible. I don't have to talk to this audience about that, I'm sure, but I just think it would be completely logical to use such an approach. You have well-validated markers of risk and a suitable preventive approach, then it's kind of a no-brainer, no pun intended.
Okay, I hope can you hear me? Okay, so the next one is due to this stigmatization of vaccination and cultural and regional differences in accepting and adopting vaccination, how can we address these disparities?
So that kind of goes to what I was saying just a minute ago. Vaccine hesitancy and reluctance to use a vaccine. That's something I personally struggled with. Anyone who so a story is when I first started working on the dark side in an investment bank in London. This was 2001, and in the UK there was well, globally actually, there was great excitement, concern about the MMR vaccine, because there was some spurious research that had been published by a guy in England. And so I had one of the colleagues in the office came and asked me, his wife was expecting a baby, and asked me, would you have the MMR vaccine for your children? And I could quite confidently say, yes, I would. And I had just had 2 of my own children and fully vaccinated. And at that point, it just struck me as such an illogical thing, where the the underlying evidence in favor of vaccines, particularly for infectious diseases, is so overwhelming. And you know, it's almost in the realms of conspiracy theory to believe that you know, the vaccines against some of these infectious agents that have been used now for decades are causing some of the more it's some of the crazy theories that are out there, let's say. So I think, you know, we've always got to go back to the heart and the strength of the science supporting vaccination approaches. And in the case of a neurodegenerative disease, I think the benefits, assuming again that safety and efficacy are demonstrated, assuming that risk can be well identified with enough time in advance, I think the potential benefits would speak volumes. And you know, we can look at the evidence of people suffering from these diseases, and if we're able to prevent some of that suffering, I think it's just, yeah, again, a no-brainer. Sorry.
How easy, complicated were the funding process and clinic pharmaceutical collaborations for this program? Were there any moments in your research where you almost had to stop the studies and trials?
That's a question about the real world, and science is wonderful, and the research that we do is important in pretty much every aspect. But the realities of this clinical developments, it is extremely expensive, and so for a clinical trial to be authorized, the regulator will want to see a large body of data to give them confidence that the experimental product you put into a subject in a clinical trial is not going to kill the patient, kill the subject, is not going to cause all sorts of serious complications. And we've seen some scary instances of trials which have begun where the first few individuals really suffered some terrible complications. So the cost of simply getting a product to the point where you can enter human clinical studies can be between $5 and $10 million. And that's beyond most academic groups' ability to afford. And so we rely then on some of the foundations to support that work, and companies like AC Immune and others, and getting investors to believe that the product you're going to take in an experimental setting into human clinical testing is valuable, has a potential use in the real world and a commercial return. That is hugely challenging on a daily basis. So you know, we have committed financing for the next year or so at AC Immune. We, like every biotechnology company, and one of the companies developing a vaccine on the table that I showed earlier on, has said that it would only take the vaccine into the next stage of clinical development with a partner. And the subtext there is they can only the next stage of development is too expensive for them to fund. And you know, this is a reality. At some point, you either need investors with very deep pockets and long belief time horizons, or you need pharmaceutical partners. And the pharmaceutical partners bring their own requirements for any of these projects. They want to control the developments, and they want to ensure that the studies are done the way they expect. But you know, they're investing $50 to $100 million for a phase 3 program, so you know, they should be able to make those requests in the development program. So it's hugely expensive, and not every program gets funded because it is not always easy to find the investors, and sometimes the pharmaceutical partners just kill a program because it's not part of their strategic universe anymore. It's sad.
Okay, thank you. We have now our last question, and then we will proceed to concluding. So will the trial also include any test to assess the cognitive evolution of the patients?
Yes. So the study that we are doing with the A-beta vaccine is looking at safety primarily, then the immunogenicity of the vaccine to ensure that we're getting the right sort of antibodies. We're also looking at the amyloid PET imaging data to evaluate the effect on amyloid plaques. And then as a series of secondary endpoints, we are looking at that battery of tests to evaluate cognition, activities of daily living, and so on. There is the MMSE scale, there is the ADAS-Cog scale, there is the ADCS, ADL. So there are a variety of the clinical scales that we will be evaluating as secondary endpoints in the study, but that they will take a bit longer. One of the reasons why the field has moved on very much in the last 2 years is the introduction of PET imaging. It gives you an objective measure of disease activity and of the activity of a therapeutic intervention. The clinical scales, all that they enable you to characterize the performance of the patient, there's a lot of noise in those scales. So it can be that the scale can give you a response on one day, which can be quite different on the next day, depending on how a patient's feels or how well they slept or what they ate the day before. So it's great to have the objective measures to evaluate disease and the therapeutic interventions. That with the cognitive scales and the performance scales, I think that's the right constellation to bring to the field.
So I was saying that it was actually a wonderful conclusion for the conference, and it was really nice to know you and your company. And so now that we know you, we will look at press release to know novelties, and let's cross our fingers that is going to improve by the time. Yeah, really.
Well, we had a press release out. We had a press release out yesterday on an alpha-synuclein tracer in Nature Communications publication. So there are a few things that will hopefully keep the scientists interested in what's going on. And there's a lot of boring stuff as well. But we try to one of the underlying themes that companies like us and others should adhere to is scientific rigor. And we try in everything that we do to ensure that that's at the heart of our developments. And with that, hopefully, we can succeed. But it's been a pleasure to participate in the conference, and I look forward to hearing the news of the next one.
Yeah, also news from scientists might be interesting.
Absolutely.
From our side. Okay, so Léa.
Thank you very much for your presentation.
I leave you the scene for the conclusion, okay? Yes. So it was a great organization and a great program, I would say. Okay? Thank you.
Thank you very much for your participation. Professor Gary, thank you very much for your presentation. It was highly informative. I would like to note how grateful we are to the partners, sponsors, speakers, colleagues, and attendees for contributing to this conference. I hope the concepts you learned and grasped throughout the past 2 days stick with you. So until we see each other again at the next Neurocon. Thank you, everyone.